Hello everyone. So the next topic which we will be discussing here is about the endocytosis. Now let's start with the endocytosis with the introduction first. So the endocytosis, it is the process by which a cell moves large amount of material or non-dissolved particles into the cytoplasm from the outside environment. So this is termed as the endocytosis. So endo, which means within, and cytosis, which means cell. So this process, as the name suggests, this process takes place within the cell. So this is the process by which a cell moves large amount of materials in the cytoplasm from the outside environment. Now, how does this endocytosis work? So, endocytosis is the case when a molecule causes the cell membrane to bulge inward and form a vesicle. So, they will form a vesicle. Now, there are three types of endocytosis. Now, what are the three categories of endocytosis? So there are three types of endocytosis. What are the three types? There is a receptor mediated endocytosis, there is a pinocytosis, and there is a phagocytosis. So first is the receptor mediated endocytosis, which is being mediated by the production of small vesicles that have a morphologically characteristic coat made up of cytosolic protein, that is the clathrin, and they are found virtually in all the cells and form domains of the plasma membrane termed as the clathrin coated pits. The next is about the pinocytosis, so which usually occurs from the highly ruffled regions of the plasma membrane, and it is the invagination of the cell membrane to form a pocket, which then pinches off into the cell to form a vesicle filled with a large volume of extracellular fluid and molecules within them. The next is about the phagocytosis, which is a process by which cell binds and internalize the particular matter which is larger than around 0.75 micro-m in diameter, such as small-sized dust particles, cell debris, microorganisms, and apoptotic cells. So these processes involve the uptake of larger membrane areas than clathrin-mediated endocytosis. So this was about the types of the endocytosis. Next, this is a receptor mediated endocytosis. So what is happening in this particular process is, so this is the first part in which the ligands, they bind to the membrane receptor. So here is a receptor which is in the blue color, and here the ligand is binding to this membrane receptor. In the next step, when the ligand is binded to the cell receptor, there is a receptor ligand which migrates to the clathrin coated pit. Now, when there is a clathrin coated pit, the next step which takes place is the endocytosis. So this is forming a vesicle kind of thing. So this is the endocytosis process. After the endocytosis process is being done, there is a vesicle which loses the clathrin coat. So you can see here, there is a vesicle which has been losing this clathrin coat. So this is about the intracellular fluid, and the processes which we were talking about was the extracellular fluid. Now, in the next step, when the vesicle, it loses the clathrin coat, in the next step, what is happening is the receptors and the ligands, they separate. So you can see there are receptors which are being separated from the ligands. So this is the endosome, and from here the receptors and the ligands, they are being separated. So in the second step, it binded. So the receptor and ligand, they binds to that. But in the fifth step, they are separating. After they separated, the ligands, they go to the lysosomes or Golgi for the processing, and the receptors, they there is a transport vesicle with receptors which move to the cell membrane. So when this transport vesicle, it moves to the cell membrane, they will fuse. So here there is a fusion which takes place, and after that there will be an exocytosis process. So this is how the endocytosis and the exocytosis process goes on in a cell. Now the components of the endocytosis. So the endocytosis pathway of mammalian cells consist of distinct membrane compartments which internalize the molecules from the plasma membrane and recycle them back to the surface or sort them to degradation. So the principal components are early endosomes, late endosomes, and the lysosomes. So these we have seen in the diagram as well. So what is early endosome? So early endosome, they are the first compartment of the endocytotic pathway. So early endosomes are often located in the periphery of the cell and they receive most types of vesicle coming from the cell surface. They have a characteristic tubulovascular structure and they have a mildly acidic pH as well. So they are primarily sorting organelles where many endocytose ligands dissociate from their receptors in the acid pH of the compartment. So this is about the early endosomes. The next is about the late endosomes, which receive the endocytosed material en route to lysosomes, usually from the early endosomes in the endocytic pathway, from trans-Golgi network in the biosynthetic pathway, and from the phagosomes and phagocytic pathways. So the late endosomes often contain protein characteristics of the nucleosomes, mitochondria, and mRNAs, which include the lysosomal membrane, glycoproteins, and acid hydrolase. So they are acidic in nature and they have a pH of about 5.5. Next is about the lysosomes, which are the last compartment of the endocytotic pathway. So their chief function is to break down the cellular waste products, fats, carbohydrates, proteins, and other macromolecules into simple compounds. These are then returned to the cytoplasm as new cell building materials. So to accomplish this, lysosomes use some 40 different types of hydrolytic enzymes, all of which are manufactured in the endoplasmic reticulum, modified in the Golgi apparatus, and function in the acidic environment. And the pH of the lysosomes is about 4.8. And by electron microscopy, usually it appears as large vacuoles containing the electron dense material. So they have a high content of lysosomal membrane proteins and active lysosomal hydrolases, but no mannose 6-phosphate receptor. So they are generally regarded as the principal hydrolytic compartment of the cell. So this is about the lysosomes. These are some of the references. Thank you.